Well, welcome to our PowerPoint about nucleic acid. This is the last biomolecule that we'll be looking at in this way. And we're pretty familiar, I think, with our nucleic acids because we've all sort of heard a little bit about DNA and what it does. And DNA, of course, is one example of a type of nucleic acid. So we're first going to look at the monomer unit that we use to build all of our nucleic acids. So our monomer is called a nucleotide, and the nucleotide is a very complex monomer unit made of 3 different parts: a nitrogen base, a pentose sugar, and a phosphate group. And if you look at the graphic here, it's sort of done out so it looks very basic here, kind of elementary way of looking at it, but it's a good way for us to see those parts. We've got our phosphate group in sort of a bluey purple. We've got our pentose sugar in a fuchsia or magenta color, and our nitrogenous base in the green. And we've got the angles here in such a way that, or in a way that we will see in lots of different kinds of diagrams, with the phosphate up, the roof of the house of the pentose sugar pointing up as well. So a little bit more detail about those parts. First, the nitrogenous base, or nitrogen base. There are 5 different bases that are used in the nucleic acids. 2 of those belong to a category called the purines, and the purines have 2 rings in their structure. And those 2 nitrogen bases are called adenine and guanine. And again, from grade 11, you might remember them as A and G. We also have another group of nitrogen bases called the pyrimidines. And the pyrimidines have a single ring structure. And there are 3 of those: thymine, cytosine, and uracil. So T, C, and U. Thymine we only find in DNA. And uracil we only find in RNA, but cytosine we find in both. So this, again, is just part of a nucleotide, the nitrogenous base that we see in the graphic at the bottom. And in the top part of the graphic, we see the purines with adenine and guanine, and we can see that double ring structure. They're very similar looking, but they're not identical. And then at the bottom, we see the pyrimidines. Again, we're seeing that they're single ring structures and that they look very similar, but not exactly the same. Okay, the second part of our nucleotide is the pentose sugar. Pentose, we recall from doing our carbs, means that they are 5 carbon sugars. And we had a little look at these in our carb PowerPoint. So the 2 pentose sugars are ribose, which we find in RNA and several other nucleic acids, and deoxyribose that we find in DNA. And deoxy, we recall, means that there is 1 less oxygen. And in the graphic here, we can see at carbon number 2, in deoxyribose on the right-hand side, we have 1 less oxygen at carbon number 2 than we have in ribose sugar on the left-hand side here at carbon number 2. And the last part of the nucleotide is a phosphate group. It's a little hard just to find a sort of random phosphate. So I've given you a graphic here of a complete nucleotide. So in this nucleotide, our phosphate group is circled in dotted sort of pink color, and we can see the phosphorus in the center and the 4 oxygen around, one of them double bonded and the 2 of them with the negative charges. And you can see here actually in the graphic that we've got a 5' just before the phosphate group, and that's telling us that this phosphate group is actually attached to carbon number 5 of the sugar. So again, look above here at the ribose and the deoxyribose, and look at where carbon number 5 is, and that phosphate group is attached to carbon number 5. So understanding the nucleotide, our monomer unit, brings us to the next sort of step in things, which is forming a polynucleotide. So we're going to take some of those nucleotides and put them together. There's lots of different ways that can happen. And in fact, the nucleotide can actually stay as a single nucleotide but add phosphate groups, and then we would have an energy molecule that we call ATP, which we'll look at later. Polynucleotides in general, so if we're taking one monomer unit, attaching it to another and another to form RNA or DNA, those things are formed, those strands are formed by covalently bonding the monomer units together. And we are basically bonding the 3' carbon of one sugar to the 5' of another sugar through a phosphate group. And that reaction is a condensation reaction similar to what we've seen when we're forming other polymer units. So the kind of linkage that forms in the backbone of a DNA or an RNA is called a phosphodiester linkage. And when we get a good graphic up here, I'll point out why that is called a phosphodiester linkage. And what happens in the backbone then is we get this repeating series of sugar, phosphate, sugar, phosphate, sugar, phosphate, with the nitrogen bases just kind of sticking out from the sugar groups. So the sugar is kind of like in the middle, if you like, between the phosphate and the nitrogen base. Exactly which nucleotide you get in what order is going to determine all kinds of things in a living system. The plan for the order of those nucleotides is basically passed on from generation to generation to generation. And again, we're looking here, or what I'm talking about here is that idea that the A's, T's, C's, and G's are all going to occur in a particular preordained sequence. So if we look at this graphic now, let's go back to the idea of a phosphodiester linkage. And I want you to look at the nucleotide that contains base number 1. If you look at the 3 carbon end of that sugar, of the structure that looks like a house there, if you look at the 3 carbon end, it's labeled 3 prime. You can see that there is a link between the oxygen of the phosphate group that's there, which is actually the phosphate that belongs to the nucleotide that contains base number 2, because you can see base number 1's phosphate group coming off its carbon number 5 above. But that phosphate group is attaching carbon number 3 of the base 1 nucleotide to carbon number 5 of the base 2 nucleotide. And if you think about an ester linkage is that single bond, the single bond holding sort of carbons together. And if you look at it sort of coming from the top down through the phosphate and from the phosphate to the right through the next carbon, carbon 3 and carbon 5 are sort of bonded to that phosphorus through what we would call an ester linkage. So we get a phospho rather than a carbon to carbon ester linkage. We have a phosphodiester because there's 2 ester linkages there that are holding them together. So that's our phosphodiester linkage from carbon 3 of one nucleotide to carbon 5 of the next nucleotide. Okay, when we're forming DNA, I know that we hear all through sort of our school experience that DNA is a double helix in its formation, and it's made from 2 polynucleotide chains that are joined together. And those nucleotide chains are joined together through hydrogen bonds. So hydrogen bonding is very important here. Remember, hydrogen bonds are intermolecular forces, physical bonds, not as strong as an ionic or covalent bond, but still the strongest of the hydrogen bonding is the strongest of those physical bonds. So what happens is you get 2 sort of parallel strands of DNA, the sugar to phosphate strands lining up, and they line up in such a way, we call it anti-parallel, and we'll look at that in a minute, that the bases, which are kind of hanging off the sugar group, are able to bond with each other, forming hydrogen bonds. So if the bases are guanine and cytosine, they will form 3 bonds, 3 hydrogen bonds. If they are adenine with thymine or adenine with uracil, they will form 2 hydrogen bonds. And you'll find that the bonding won't occur in any other way. So we're always going to have one purine and one pyrimidine bonding together. We're not going to have 2 purines bonding with each other or 2 pyrimidines bonding with each other. So if we look at this graphic, we can see at the top of the graphic we've got a thymine bonding to an adenine, and we've got our 2 hydrogen bonds forming there, represented by the dashed red lines. And under that we have a cytosine and a guanine, and we can see in that case that we have 3 hydrogen bonds holding them together. On the left-hand side of the diagram is one strand, one polynucleotide that was part of the DNA. And again, we can see that phosphate sugar linkage. And on the other side, on the right-hand side, we can see the same thing happening where we've got a polynucleotide and we've got phosphate sugar. You may notice, however, that the sugar molecules on either side of that chain, so the left-hand chain and the right-hand chain, are opposite to each other in their orientation. So on the left-hand chain, the roof of the sugar, if you like, where the oxygen is right at the top, is pointing up, but the sugar on the other side appears to be pointing down, and that's very important. If those strands were not lined up that way, then the hydrogen bonds would not form between the nitrogenous bases, and the 2 strands of DNA would not be held together. So using that same diagram again, I'll sort of identify that situation where we've got the sugars on one side oriented in one direction and the sugars in the other side oriented in the other direction as anti-parallel. We call that an anti-parallel arrangement. So they're parallel, the base strands, or better not use the word base that way, but the strands of sugar phosphate are parallel to each other, but the orientations themselves are opposite. And we use the numbering on the carbon of the sugar to tell us which way a particular strand is oriented. And when we're talking about a DNA chain, DNA double helix, we will say that one chain is oriented 3' to 5' and the other one is oriented 5' to 3' in order for the hydrogen bonding to occur. In this graphic, even though it's a little blurry, we can see multiple examples of the purine pyrimidine setup where we have double 2 hydrogen bonds or 3 hydrogen bonds. But what I wanted to point out on this was the 5' end of one strand is at the top, where the 3' end of the other strand is at the top, and vice versa at the bottom. And again, what we're seeing here, if we look at the left-hand side of this picture where the sugar roofs are pointing up, it's carbon number 5 that is sort of at the top. So carbon number 5 is reaching up toward the top of the diagram and is attached to his own phosphate group. Where if we look on the right-hand side, we'll see that the 5' end of the carbon is actually pointing down toward the bottom of the graphic. So the roof of the sugar is pointing down, and so carbon number 5 on that sugar is pointing down, and we can see that it's attached to its phosphate group pointing down. So these 2 strands are anti-parallel to each other, allowing the hydrogen bonding to occur in the middle. All right, we already mentioned that not all nucleotides are going to be forming polynucleotides such as RNA and DNA. Some of the nucleotides are also used for energy. So those polynucleotides tend to be for storage of information, where the examples we're going to see on this slide are going to be examples of energy nucleotides. Often will be used to transfer energy. So we're often seeing energy sort of being shifted around using these molecules as intermediates. So ATP is the one we're probably most familiar with, adenosine triphosphate, and we called it earlier in the class our energy coin. And we've seen this graphic before. We've got our nitrogen base over on the left-hand side in the blue, and the nitrogen base is adenine in this case. We've got the ribose sugar in the middle. So we see, looking down at carbon number 2, we can see that it's an OH group there. So it is ribose, not deoxyribose. And in orange, we can see our 3 phosphate groups. And we've mentioned the idea that these are very good energy molecules because of all the negative groups that are there. We get a lot, or it takes a lot of energy to keep those together. And so it's a good little storage place for energy. Another energy molecule that we haven't looked at yet is called NAD, and we'll be seeing this molecule again when we do cellular respiration. Nicotinamide adenine dinucleotide. Maybe I should do amides next time because there's an amide in this particular group. So the actual structure of the thing is called a dinucleotide, and this one is quite obviously a dinucleotide in the sense that we've got 2 sugars, we've got 2 nitrogen bases, and we've got 2 phosphate groups, and they're sort of held together in the center with one of the oxygen groups. And this molecule is going to be very important for us when we're moving electrons around in cellular respiration. FAD is also a molecule that we use in cellular respiration. This one is flavin adenine dinucleotide, and it doesn't look quite as obviously as like a dinucleotide in the sense that we've only got 1 sugar, but we've got 2 sets of nitrogenous or nitrogen bases, and those 2 phosphate groups holding them together again. It's just that the top nitrogen group, if you like, doesn't have a ringed sugar, but has several, we've got more like a linear sugar group there, so not quite as obvious. Both of these, NAD and FAD, are both used in cellular respiration, so we'll see both of those when we do our metabolism unit next. And something I will point out to you here, next time you're eating cereal and you're reading that little panel on the side, have a look and see if you can find niacin and riboflavin, both of which are vitamins that are often found in breakfast cereals. And nicotinamide adenine dinucleotide is derived from niacin, and FAD, flavin adenine dinucleotide, is derived from riboflavin that you see in that ingredients list. The last one that we're going to look at is NADP, and very similar to NAD. However, we've got an extra phosphate group on there. So nicotinamide adenine dinucleotide phosphate, and in this case is used in photosynthesis, not in respiration. And we've got if we have a look at it here, we can see our 2 ring sugar groups. We can see that 2 phosphates in the center. We can see our 2 nitrogen rings. And then on the bottom, carbon number 2 of the sugar to the right, we can see another phosphate group. So that's our sort of extra phosphate group that we're looking at. And those are our energy intermediates that we'll be looking at this year. So we've come to the end of our presentation on nucleotides. Make sure that you write down any questions you have for class to ask to clarify anything.